The history of Earth began 4.6 billion years ago, a time when everything was vastly different from what we know today. Our planet looked alien and unknown, yet it would eventually become hospitable enough for life to flourish and for humanity to permanently settle. How did the world around us form, and how did it evolve to become the Earth we know? Through a series of 4 videos, we embark on an extraordinary journey, tracing the history of our planet since its birth, diving deep into its core from its formation to its long evolution. Today, we begin episode 2. We've already covered Earth's formation up to 3 billion years ago. We'll continue this journey all the way up to 485 million years ago. Every planet has a history. This is Earth's. Our story picks up with the Pongola glaciation, which began 2.9 billion years ago. This was the first ice age in Earth's long history. This period is defined by the presence of at least one ice sheet, linked to a significant drop in global sea levels. When continents drifted into polar positions, snow piled up even during summer. Simultaneously, mountain building exposed fractured rocks, which chemically reacted with the atmosphere, consuming large amounts of carbon dioxide. This weakened the greenhouse effect, causing global temperatures to plummet. Earth's albedo, or reflectivity, increased dramatically. As half of the planet's surface froze, nothing could stop the runaway glaciation, creating the infamous snowball Earth appearance. Around 2.7 billion years ago, a new supercontinent began to form, Kennerland, including parts of Western Australia and the Kalahari. Massive magmatic eruptions occurred beneath the oceans, peaking with the explosion of komatiites. Volcanic arcs rose from the mantle, followed by mountain building, granitoid settling, lithosphere stabilization, and collisions with other cratons. All these events shaped Kennerland above sea level. About 2.5 billion years ago, plate tectonics reached a turning point. Until then, plate movement was both horizontal and vertical. Vertical movement, or sagging, occurred when dense rocks sank beneath less dense crust, creating depressions and contributing to sedimentary deposits. As the mantle's composition changed, komatiite formation slowed. Vertical tectonics ceased, leaving only horizontal motion. During this time, a heavy, unbreathable atmosphere dominated Earth. Methane-producing microorganisms were replaced by photosynthetic organisms, releasing vast amounts of oxygen. The world resembled Saturn's moon Titan, veiled under a thick orange haze. Cyanobacteria flooded the seawater with oxygen, which eventually reached the atmosphere. This chemical evolution caused the Great Oxidation Event, a mass extinction as oxygen poisoned anaerobic organisms. Survivors evolved protective mechanisms, eventually giving rise to aerobic life forms. Oxygen provided new energy sources and reshaped ecosystems and geological interactions. Around 2.4 billion years ago, Earth entered one of its longest ice ages, the Huronian glaciation. The emergence of photosynthetic life reduced methane, a potent greenhouse gas causing temperatures to drop. Carbon dioxide trapped in limestones further cooled the planet. For tens of thousands of years, temperatures fell by as much as 50 degrees Celsius, freezing water globally. Albedo increased, accelerating cooling in a runaway effect. Volcanic activity intermittently released carbon dioxide, temporarily warming the planet. Over 300 million years, cycles of glaciation and thawing occurred 4 times. The retreat of glaciers triggered massive erosion, breaking down silicates and precipitating carbon into oceans. Cratons accumulated into larger land masses, and oceans gradually deepened. Iron in primitive oceans existed in a soluble green form. With oxygen production, iron oxidized into reddish ferric iron, precipitating to the ocean floor as sediment. During glacial periods, oxygen slowed, producing gray or greenish layers. Once iron precipitated, oceans cleared, allowing sunlight to penetrate deeper. Coccoid bacteria or phytoplankton thrived. These tiny organisms performed photosynthesis, converting carbon dioxide, minerals, light, and water into organic matter. Phytoplankton became primary producers supporting other life forms. Driven by plate tectonics, a new supercontinent formed around 2.2 billion years ago, Columbia, also called Nuna. It contained most of Earth's continental crust, stretching 12,900 kilometers north to south and 4,800 kilometers wide. Continental blocks collided over millions of years, forming mountain belts and fusing cratons from around the globe. Columbia remained intact until 1.6 billion years ago, when it began breaking apart. With oxygen now available, life became more complex. Cells learned to use oxygen, and respiration emerged. Eukaryotes, cells with a nucleus and organelles, appeared around 1.45 billion years ago, with some traces of multicellularity extending to 2.1 billion years. These cells were larger, contained linear chromosomes, and carried organelles like mitochondria. Acritarchs and multicellular fossils in Gabon provide early evidence. Mitochondria arose from endosymbiosis, anaerobic cells engulfed bacteria capable of converting energy efficiently. These bacteria became organelles, powering the eukaryotic cells. From 1.8 to 0.8 billion years ago, Earth entered the boring billion. Tectonics and atmospheric conditions were stable. Oxygen remained low, and evolution progressed slowly. Though uneventful, this period laid the foundation for explosive events in the next billion years. As the planet emerged from its global freeze, a newly formed ozone layer set the stage for the Ediacara fauna. This critical atmospheric development allowed for the proliferation of these early large organisms, which thrived in the oceans as flattened discoid forms. These early large organisms, though they might superficially resemble jellyfish, were fundamentally different in their biology and lifestyle. They lived in dense communities, firmly anchored or resting on the seafloor, forming extensive ancient marine ecosystems. Mevsimatids, for example, were a fascinating group of ediacaran organisms. They possessed discoid bodies that could reach an impressive 12 centimeters in diameter, and uniquely, these bodies contained internal sand-filled tubes, the purpose of which is still debated by scientists. Vendobionts, a diverse group of ediacaran life, included organisms like rangiomorphs. These creatures had distinctive sac-like bodies that were partially buried in the soft sediment of the ancient seafloor. Rangiomorphs were particularly notable for their fractal and self-similar structures, resembling fern fronds anchored to the substrate, and they dominated deep-water ecosystems for millions of years. One iconic family was the Dickinsonids, which included the well-known Dickinsonia. These flat, oval-shaped animals exhibited a wide range of sizes, growing from just a few millimeters to an impressive 1.4 meters in length. Their unique segmented bodies are found in fossils across Australia, China, Russia, and Ukraine, suggesting a widespread distribution. Lacking mouths and traditional digestive systems, Dickinsonia and similar organisms developed a unique feeding strategy. They absorbed nutrients directly from the rich bacterial mats that covered the seafloor, essentially grazing on these microbial carpets for sustenance. Other forms of ediacaran life included yorgids, which were observed to crawl slowly across the sediments, leaving behind distinctive trails. Another striking organism was Charnia, which had large leaf-like structures anchored vertically in the sand, some reaching up to 2 meters in height. These Charnia organisms often grew in dense clusters, creating what paleontologists describe as small thickets on the seafloor. These underwater forests provided complex habitats and likely influenced water flow and sediment deposition in their ancient marine environments. The trilobizoans were a particularly rare and unusual group among the Ediacara fauna. They were characterized by their unique tri-radial symmetry, featuring 3 distinct, spirally twisted lobes and intricate radial grooves covering their bodies, setting them apart from other Ediacaran forms. Microbial mats were absolutely crucial to the entire Ediacara ecosystem, forming the fundamental base of the food chain. These vibrant, earthy green layers of microorganisms provided the primary energy source for much of the Ediacaran life. The feeding strategies within the Ediacara ecosystem were diverse. Some Ediacara animals fed directly from these abundant microbial mats, while others evolved to filter microorganisms and organic particles from the surrounding seawater, showcasing early ecological specialization. The Ediacara fauna thrived for millions of years until their mysterious disappearance around 542 million years ago. Theories for their extinction range from significant environmental changes and increased competition with emerging Cambrian species to potential ozone depletion caused by shifts in Earth's magnetic field, with some studies suggesting decreased global oxygen availability as a key factor. Following the monumental breakup of the supercontinent Rodinia, the southern hemisphere became dominated by 4 major continental blocks. This geological rearrangement profoundly influenced ocean currents and the distribution of shallow marine environments, setting the stage for new life forms. These 4 major continental blocks included Laurentia, which encompassed what would become North America and Greenland. Siberia lay to the east of Laurentia, while Baltica comprised the landmasses of Scandinavia and Western Russia. Gondwana, the largest of these blocks, was a vast supercontinent spanning from modern-day South America and Africa to Australia, India, and Antarctica. As these continents drifted, shallow seas began to spread extensively across their continental shelves. These vast, sunlit, and nutrient-rich shallow marine environments created ideal habitats for the rapid diversification of multicellular animals, known as metazoans. These early metazoans represented a significant evolutionary leap, as they were characterized by differentiated cells organized into tissues and possessed complex internal structures such as nuclei and specialized organelles like mitochondria, enabling more complex biological functions. Around 541 million years ago, life on Earth underwent a monumental and rapid diversification event known as the Cambrian explosion, often dramatically referred to as the Big Bang of life. This period marked an unprecedented burst of evolutionary innovation. During the Cambrian explosion, new species and entirely novel body plans appeared at an unprecedented pace. Benthic animals, those living on the seafloor, rapidly developed complex feeding strategies, leading to food chains that became far more intricate and interconnected than anything seen in the preceding Ediacara period. Some scientists propose that significant shifts in Earth's rotational axis played a crucial role in this diversification. These shifts are thought to have moved continents to warmer, more equatorial latitudes, which in turn led to a substantial expansion of shallow seas and a dramatic acceleration of biodiversity. From 541 to 485 million years ago, a series of distinct faunas emerged, each providing a snapshot of the rapidly evolving life forms. These included the Tamat, Chungjiang, and Burgess faunas, each contributing unique insights into early animal evolution. The Tamat fauna, named after the Tamodian stage of the early Cambrian, consisted primarily of tiny fossils, typically measuring only 1 to 3 millimeters long. These minute remains included early forms of mollusks and annelids, providing crucial evidence of early animal diversification. A key evolutionary development observed in the Tamat fauna was the display of shells composed of calcium, silica, and phosphate. This marked the first major stage of skeletal mineralization, a groundbreaking innovation that provided protection and structural support for these early animals. Known collectively as the small shelly fauna, these fragments represent a pivotal moment in evolutionary history. They either signify the very beginning of hard-shelled animals, providing a protective advantage, or they are the remains of fragile species that quickly went extinct, highlighting a period of intense evolutionary experimentation and rapid turnover. Archaeocyaths, early sponge-like organisms, built reefs up to 2 meters high, providing the foundation for future ecosystems. They had dual cone-shaped skeletons of magnesian calcite, lived on the seabed, and filtered nutrients from strong currents. Next came the Chengjiang fauna, preserving at least 16 phyla and 196 species, 40% arthropods. Exceptional mud preservation allowed us to see both hard and soft tissues, including digestive and nervous systems. Among them was Hyquechthys, the first true vertebrate. Measuring just 2.5 centimeters, it had a bony skull, gills, and V-shaped myotomes for agile movement. Its skeleton protected vital organs and allowed independent head movement, a revolutionary step in vertebrate evolution. Other Cambrian marvels included Anomalocaris, a large predator with clawed appendages and ringed teeth, and Opabinia, with 5 eyes and a grasping proboscis to collect food from sediments. At 505 million years ago, the Burgess fauna marks the height of the Cambrian explosion. Preserved in oxygen-free mud flows, it included 120 genera, 33 hard-shelled, 87 soft-bodied. Arthropods alone had 24 distinct body plans, only 4 of which survived. Arthropods dominated. Their segmented bodies, jointed limbs, and exoskeletons required molting to grow. Branchiocaris, a bivalved swimmer with clawed appendages. Morella, a soft-bodied arthropod with a horseshoe-shaped cephalic shield. Trilobites, nearly 19,000 species, with compound eyes made of thousands of calcite lenses, giving almost hemispherical vision. Hallucigenia was a worm-like oddity with 7 pairs of clawed tentacles and dorsal spines. It walked on tentacles and used spines for defense, with sensory tentacles near its head. Sponges circulated water to feed and remove waste, regenerating if damaged. Sea lilies, with their rooted stems and mobile arms, trapped plankton while defending themselves with calcified bodies. Brachiopods, like Lingulella, filtered plankton with internal tentacles and absorbed oxygen, an early example of efficient organ systems. By 485 million years ago, life was ready to conquer land. Deserts were empty with no vegetation. Early plants, moss-like and lichen-like, colonized rocks. Without fully developed vascular tissues, they endured cycles of drying and rehydration. To survive, they evolved cuticles to reduce water loss and stomata for gas exchange. These pioneering plants paved the way for soil formation, setting the stage for future terrestrial ecosystems. And that brings us to the beginning of life on land. If you're curious about what Earth looked like 500 million years ago, don't forget to like and subscribe. Join us next time as we continue to explore our planet's incredible history.